Sí, sí se oye. Ok. Sí. Ajá. Ok. Aquí. ¿Qué habla ahí? Ah, ok. So, hello everyone. Please take a seat. We will start the next session and introducing Carlos Hiller, please. Woo woo! So, Carlos Hiller is a plastic artist born in Argentina and naturalized Costa Rican with 30 years of residence in this country. His great passion for the ocean and diving led him to specialize in portraying the underwater world. Through public morals and environmental education, it constantly disseminates the need to protect the oceans, both in the country and abroad. His artistic works are present in private collection through the world, being cataloged by specialized media as a global reference in underwater art. Carlos even paints underwater, being one of the few artists in the world who take art to such an extreme. So, thank you.
Thank you very much. First, thank you very much for allowing me to be here, for allowing art to be part of this Congress, and also for including art in these scientific expeditions. I know that the space in the boats are vital, are very important, and the effort of having not only one but two artists, also Michael Rowe, were there. Actually, he's coming to tomorrow, and he's going to be exhibiting artwork on the art exhibition. I hope also we are doing good work for showing the world what is through our art, what is underwater, about all this fantastic ecosystem. But instead of talking, I prefer to paint. That is my better language. So it's going to be about 10 minutes, 11. Hope you will enjoy the music also, which is the music from my good friend Manuel Obregón, Carlos Tapado Vargas. Thank you very much. Gracias. I was thinking for many months, actually almost a year, it took me to realize the way I can send the urgent message, not only protection, but also what can motivate this protection. And I found a key word for me that is fragility. And I came out with this idea of creating these mobile sculptures, which talks all about fragility and equilibrium. It looks very simple, but for taking this equilibrium, it takes many hours of calibration. And if it moves the wires just a little bit, they are going to fall out. The sculpture itself, especially this one. Every time I move from one place to other place, I need to calibrate, and sometimes it takes hours of calibration. So I think it's a good example of how long took to these very fragile ecosystems to take this equilibrium. Just for us to be in this specific moment where we have the technical ability, the technology to get to know these ecosystems and to interfere also with this ecosystem, and also to allow the exploitation of these ecosystems. So that's the reason I put in this live painting presentation a hand representing us. It's in our hands to make a change and to allow and to or not the commercial exploitation, and to get people to know, for them to love these ecosystems, and then protection comes. You already know all of this, but this is my perspective from the artist view, from an artist. So thank you very much for your patience and for allowing me to be here. Gracias.
Thank you so much, Carlos. It's a beautiful paint. So, meanwhile, someone has a question for Carlos? A comment? Okay. So, thank you. Yes? Okay.
Carlos, thank you very much. I talked with Carlos, after the first deep sea expeditions that I did, and I knew his art, and I showed him pictures and videos from those expeditions, and saying, "Look, this is the next step into the deep," and he said, "Yeah, it's interesting." And we also met with Manuel Obregón, the guy that did the music, and talk about it, but still it was going slow, and it was great that he could be in the Octopus Odyssey expedition and see it live, and it's been a change. He's one of us now. Thank you. Gracias. Yeah.
When you start this, do you have any idea of what you're going to paint?
Sí,
Like, have you thought about it?
actually, this presentation with it changes, of course, I change every time. This is, I think, the third time I did at the Immersive Congress, something very similar, and I did also something very similar at the exhibit at the Parque Marino del Pacífico. Actually, part of the campaign Costa Rica Reconocida, we are doing all over the country murals and art exhibitions and all type of activities for growing the knowledge about this environment. So hopefully soon we're going to change the name. It's not going to be anymore Costa Rica Desconocida, but the Costa Rica that's also to be part of Costa Rica.
I guess I meant, like, when you approached the day, did you days before out, or have you thought about what you have?
Just 5 minutes before. It's always the same. Yes, yes. Just 5 minutes before, I very excited, and I decided what to paint and also what to say.
Thank you so much. We can leave here. No? Okay, so we wait some minutes. Sí, con hay un motorcito. Ajá.
Yeah, I just made this mug 5 minutes ago.
Uy.
I mean, I could do it as like an interpretive dance or something. Yeah, I know, it's all down now.
So the next speaker is Rachel Lauer. She is a geoscientist at the University of Calgary since 2015, specialized submarine geohazards, including earthquakes and landslides. She holds a PhD in geoscience from Pennsylvania State University and Masters in Environmental Engineering Geoscience from Radford University and a Bachelor in Psychology from Bryn Mawr College. Her career spans academia and applied geoscience, geophysics consultancy. Rachel's research emphasizes submarine hydrogeology, particularly fluid movement in oceanic crust. Her work has led her to conduct deep sea research, providing her with valuable insights into oceanic geoscience and its interplay with marine biology. So,
Awesome. Well, thank you. And as was stated, I think by Gus, it's going to be a hard act to follow. So, I know that everyone's favorite subdiscipline in all of the cruises that we've participated in is heat flow. So, yes, thank you. So I am going to be talking today. The title is a bit of a joke, but it's sort of following an observation that we've made sort of anecdotally in many locations, which is that the fluids that are coming out of the crust tend to be warmed up, enhanced in chemistry and potentially microbes, and they tend to be places where octopus and other critters thrive in these biodiversity hotspots. So the only biology we'll see in this entire talk is on this first slide, so moving past that. Uh-oh. Oh, here we go. Okay, so I'm going to skip past this because I think lovely introductions to this area have already been provided by many of the people that came before me, but we're going to be talking today about seamounts off of Costa Rica. And before I start talking about some earlier work that was done that sort of set the stage for the work that I'm doing now, I think it's important to understand why fluids in the ocean crust are so important as a global process. And I want to start by just pointing out the magnitude of fluids that are residing within what I call the aquifer, which is the upper part of the oceanic crust. This is made up of mostly pillow basalts. This is a very permeable part of the crust that allows fluids to be transmitted and exchanged between the ocean above and the crust that sits below the sediments. So in terms of volume, the ocean crust is hosting about 20 to 30 million cubic kilometers of fluids. Okay, so this makes it rivaling glaciers and ice sheets in terms of global storage of fluids on our planet. And obviously it's an aquifer in the sense that it's permeable, but obviously no one's drinking that water. Okay, but in terms of volume, this is ginormous. So we know that water is not static. It is not just sitting there hanging out within the crust, and that's basically the root of what I study, is the movement of that fluids both into the crust and out of the crust. That process is known as hydrothermal circulation, and a cartoon of that is shown here. Okay, so when we talk about ridge flank, we're talking about something that was introduced by Marino. Ridge flank just means that it's crust that's about 1 million years old or more, and it has been well sedimented, so the only pathways provided for the ocean to exchange with fluids in the crust itself are provided by these seamounts or outcrops that stick up above these relatively impermeable sediments below. What's shown above the top there is a heat flow profile between a recharge site and a discharge site on the right. And what you can see is the entire heat flow is depressed relative to what it should be based on the age of the crust. So the crust forms at the mid-ocean ridge, it cools through time and densifies, which was discussed earlier. And based on a regional reference and the age of the crust and some fancy math and models, we know how much heat should be coming out. In fact, it's generally lower than that wherever we measure it because of this process. So you have cold, dense bottom water making its way into a recharge site, ultimately traveling and convecting and picking up heat and solutes and potentially microbes on its way to that discharge site. And the discharge site is indicated by higher heat flow, the recharge site by lower heat flow. And this is called the hydrothermal siphon because it is self-sustaining. Okay, so this will continue to function as a siphon for millions of years as long as there is a difference in temperature between those sites, which drives pressure and flow between them. Okay, so the strength of that siphon varies depending on many things, but the siphon is thought to be ubiquitous everywhere we have seamounts. So in terms of rates, so now we're talking about movement of fluids, not just them sitting there. The actual rates of this process globally on ridge flank, so crust that's more than 1 million years old, is between 2,000 and 20,000 cubic kilometers per year of fluxes. Okay, so what does that mean in terms of things that we care about? Well, it means that this process is recycling the ocean every 100,000 to 500,000 years. As a geologist, that is a very short time scale. I know to the rest of us it seems very long, but that means since the dinosaurs, this process, has recycled the ocean like 100 times. Okay, and then thinking about it on a smaller scale, how often is the actual ocean crust being recycled? That process is happening every 1,000 to 10,000 years. Okay, so the implications for ocean chemistry and all sorts of processes that we care about is huge. The global loss as a function of our planet just cooling since its formation is 40 to 45 trillion watts or terawatts. And of that heat loss, 75% of it is happening at the ocean and ocean-water interface. And of that 75%, 30% is extracted by this process of hydrothermal circulation. Okay, so this is globally significant process. It's removing more heat than any other physical process on the planet. So going back to Costa Rica, which is why we're here, back in 2001, 2002, a series of surveys of this area were done to not just map what's there at the seafloor, but actually conduct seismic surveys and heat flow measurements, which are the dots shown on the right-hand side, with the colors basically going from purple, which is low heat flow, all the way up to bright red, which is very high heat flow. And again, higher heat flow generally indicates a place where water is coming out, lower heat flow, places where fluid is going in. And of these 11 outcrops, it was identified that about half are recharging, half are discharging, but in this gray area shown on the right, 70 to 80% of the heat that should be coming out is missing. Okay, so missing in the sense that it's been washed away by very rapid hydrothermal circulation. That is, again, the only process that can remove that much heat. The only way to justify that is having extremely high flow rates we'd never measured before in the ocean crust. And the question is, what are those rates, and are they reasonable based on what we know about Earth processes? So again, just zooming in on the pair that we really care about, we think fluid is going in, or we know fluid is going in Tangosed, and we now know it's also coming out of Tangosed. So there's a little bit of local hydrothermal circulation happening because it's a large outcrop. But some of that fluid is also making its way all the way to Dorado. Okay? And this has very high heat flow, over a watt of heat flow over Dorado. And again, this is just put in the context of that region about the size of Connecticut, which I should have come up with something in Costa Rica, but where all that heat is missing. So to kind of understand what permeability and number or spacing of outcrops would allow us to mine that much heat out of the crust, I use what are called numerical models, math, to try and answer that question. So what parameter space actually achieves that heat loss that we measured around Dorado outcrop and around this entire region? So rather than talk about the 27 different types of models I constructed, we're going to focus on one. That's 20 kilometer by 20 kilometer outcrops on the corners, spaced approximately the same spacing as Tangosed and Dorado. And the questions I'm asking are, what are the flow rates that occurred under these conditions? And how do those actually translate to things that people in this room care about in terms of flux of solutes, transport of nutrients, et cetera? Okay, so this is a finite element model that I use and constructed to simulate those conditions. On the corners, we have seamounts that are essentially the size of Dorado for these simulations. The sediment thickness was designed to emulate the 450 meters of sediment that we have within this region. And the crustal aquifer permeability was the number we're really trying to figure out, to pin down exactly what are the hydraulic parameters needed to match those conditions. And then below, we're putting in 110 milliwatts per square meter, which is what we should have coming out based on the age of the crust, which is about 24 million years old. So look at those early results. We can see this is one of the simulations that achieved and matched those heat flow based on the measurements done in this area. You can see this sort of tongue of chilled area at the base of the aquifer. There's really no flow through the sediment, which is good because there shouldn't be. It's very impermeable. And overall, the global heat flux in this area is significantly reduced because of that vigorous advection between the recharging outcrop on the left here and the discharging outcrop on the right. So looking at this in a top-down view, this is just a contour map of the simulation results for a progressively more permeable aquifer. So on the left-hand side, this is showing an aquifer with permeability of 10 to the negative 11, an order of magnitude higher, and an order of magnitude higher, further to the right. And in each case, you have a recharge site in the top left, discharge in the bottom right. And what's shown here is heat flow calculated from the model results as a result of, at the end of the simulation. And what you can see from histograms, which represent every node at the top of the model, with 10 to the negative 11, we are about 100 milliwatts per square meter, so almost no heat has been removed from this system. At 10 to the negative 10, it's getting a little bit closer to what we actually measured, which is shown in gray here, around 30. And it requires actual permeabilities of 10 to the negative 9, which is 1,000 Darcys, to actually match the measurements of heat flow that were estimated around Dorado. So I'm going to go to another cross-section, because one of the important things is you don't model something in 3D unless it's actually 3D. And to look at how 3D this is, we have to look at a cutaway here. So this is really rapid inflow happening at the recharge site, and the actual size of the vectors here reflects the velocity. So larger arrows, larger velocities. Okay, so very rapid inflow, and you can see this deeper flow towards the discharge site, which is shown to the right, but there's also arrows pointing back the other way. So you actually have counterflow happening in the shallow aquifer. Okay, so strong lateral flow, but some counterflow is happening, so it's at least two-dimensional. So you have a large, flat convection cell happening. But then, if you actually peel the sediment away and plot those same arrows, except now we can see the arrows within the aquifer itself, what we see is it's entirely three-dimensional. You have that rapid flow towards, in the deep sections of the aquifer, towards the discharging outcrop, the counterflow in the shallow aquifer, but what you end up getting is almost a dipole-type shape that is reflecting both the recharge and discharge are three-dimensional. So to answer those questions, and then hopefully get to 2 slides that I hope I can get to, that show some new results and also some implications for this in the realm of deep sea mining protection. So to answer the first question, what are the conditions required to add back 70 to 80% of lithospheric heat? Really high permeability. So the highest permeability ever estimated in the ocean crust until this study was 10 to the negative 10. We are estimating 10 to the negative 9. What are the flow rates that exist under those conditions? Well, specific discharge is kind of analogous to velocity, and under these conditions, that means specific discharge rates of up to 500 meters per day between outcrops. Okay, so this is about 3 times faster than you experience between baby bear and grizzly bear, just for context. The fluxes are 1 to 3,000 liters per second for a single outcrop. And this is consistent with previous hypotheses for this specific region based on heat flow. And then why does this translate into things that everyone here cares about in terms of flux of solutes, nutrients, et cetera? For a simple example, this would be 50 to 150 millimoles per second per outcrop of dissolved organic carbon and nitrate. In annual amounts, that amounts to 1.5 to 4.5 megamoles per year per outcrop. And again, this is a simplification. It is a model, but in terms of fluxes that are required to achieve what is observed based on the hundreds of heat flow measurements, these are the solid estimates. Ideally, we would like to test the hypotheses that arise from the simulations that were conducted, and we can do that if we think about conducting a tracer test if we had 2 outcrops that were a little bit closer together. Oh wait, we actually do have 2 outcrops that are closer together, so I'm going to skip to that one to make sure I get it because I'm on 2 minutes. As you all know, we actually focused, other studies were focused further to the north. We actually focused on La Pulperia and Caballito, which are very close to the transition, but had not previously been seen with human eyes or densely populated with heat flow measurements. And it turns out that these new data obtained last year identified the 3rd global pair of recharging and discharging seamounts, and we're conducting a modeling study right now that has preliminary results that I'll wait for my grad student to present those the next time we see each other. So here is a glimpse of just the heat flow data plotted alongside of, this is just the topographic data in terms of elevation. On the left is Caballito, on the right is La Pulperia, and you can see that heat flow is generally depressed between them and elevated. This is the same pattern we saw with Dorado and Tango Sed in the 2001 and 2002 work and in 2013. And so like that other pair, the missing heat, in this case, our heat is right around that gray bar. That's the same very low heat flow that we've estimated in other parts of this system. So again, we anticipate very high permeability and the potential for a tracer test. If you're getting 500 meters per day between these two, you could actually, if you can locate a place to put the tracer in, monitor it in a single expedition, which is a really cool opportunity. And then in thinking about this in the context of establishing and expanding marine protected areas, I've started working with lots of Canadian biologists, thanks to the expedition with you all, to try and protect larger regions of the seafloor by acknowledging the hydraulic connection between these pairs of seamounts. So if we use your favorite measurement, heat flow, to start mapping where water's going in and where it's coming out, we can actually double or triple the size of the parts of the seafloor that we're protecting by acknowledging that hydraulic connection between them. Because you also can't mine or disturb the area where the fluids are going in without impacting the biology where it's coming out. So, that's it. I think it's on that time. No,
No.
that one's on.
Hello. Okay.
Hey.
Yeah. Thanks, Rachel, for this very illustrative talk. So once, just in terms of mass balance, once you get steady state, you assume that whatever goes in goes out. So if when we look at Tengosed as a huge mound
Yeah.
compared to El Dorado,
Yep.
so you make you think that there's more water getting in than the water that goes out. So somewhere the balance has to be established. So what you feeling about it?
Yeah, so I mean, the advantage of doing the simplified simulations, what I do is you can use symmetry to actually simulate a field of outcrops. And you're absolutely right that, there's two complications there. One is that some of the water that's going in Tengosed is coming out of Tengosed. And we saw that a kilometer away from the skate park and skate nursery. And that is also revealed through the physical simulations. So when you have that much surface area available, some of the fluid that goes in is coming out, and some of it is going into the crust. And you're right, the question of where it's going after it gets there, if you're simulating a pair, is not the same as if you're simulating four, because you could have a series of dipoles, basically, that are intersecting, and that means that one outcrop may be pulling from multiple recharge sites. Yeah. So there is, and in terms of mass balance, yeah, that would be a tough exercise to do without having really good numbers on what exactly the fluxes are into or out of. Yeah. But this gives you like a ballpark order of magnitude. Yes?
This might be a really simple geology question. Why is it in the pairs? Why is it that the larger sites act as the recharge sites and the smaller ones act as the discharge sites?
No, that's actually a physics question.
Oh, okay.
So very good. So it is literally, okay, so what ends up sustaining a siphon is, you have one outcrop here, one outcrop here. This guy's cold, this guy's warm. Cold water is denser, warm water is less dense. So the pressure at the base of this one is higher than the pressure over here. So it drives flow. Okay, so that's how this siphon works. But also, the bigger surface area is harder to maintain a specific temperature. When all the heat is going towards the little guy, and that one gets really warm, it stays warm, and it sustains the siphon. So it generally favors the small outcrops being the discharge sites. But in the surface area is what generally facilitates enough water getting in to sort of start that process. Yeah, so it's physics, actually, not geology. Yeah.
Thank you so much for this nice presentation. So we will continue with Gustavo Ramirez. Gustavo Ramirez, he's an assistant professor at the California State University in Los Angeles, where leads an ecogenomics and microbial ecological lab. His current interests include using large-scale data analysis to understand the coevolution and geological and biological processes, rock micro interaction, determining the ecological underpinning. Sorry, yes. My fault. Julie Huber. Okay, so. Okay, no, it's fine. Okay.
Okay, yeah, if we could go back to the first slide. So hi everyone, it's really fun to be here. My name is Julie Huber. I'm a senior scientist at the Woods Hole Oceanographic Institution. Beth showed a map of the east coast of the United States. I'm south of her, a place called Cape Cod, surrounded by the ocean. One slide back. And I'm really happy to be here. This is the most time I've spent in Costa Rica besides your waters. So I'm really happy to be here and appreciate the invitation. So I will just start with this slide. I'm going to be talking about microbial ecosystem services in the deep sea using Pampa Submarina as a test case scenario for examining these things. But I want to remind everybody that the deep ocean provides all of these essential ecosystem services to all of humanity. So ecosystem services is a term that has caught on in popularity. I don't know for how much longer in the United States, but basically these are services that provide from nature that provide services to humanity. And it's kind of a hard thing to connect your brain to, but it's really important, especially as we think about stewardship, resource extraction, and our connection to the natural world. So in the next slide, and I believe I'm in charge of this now, sorry about that. Do I just press play? Yeah, okay. So I think it's pretty easy when people live by the coast to connect with their natural world, but over the last 5 years or so, a lot of us have been trying to help people understand the essential ecosystem services that the deep ocean provides. So this was an infographic that Josie put together about these deep ocean ecosystem services. And we just heard, the volume of the ocean is incredible, not only also within oceanic crust, but I want to focus today on what's happening at the bottom, at and beneath the seafloor, and I want to talk about it from a microbial perspective. So, and ask this question: what services do microbes provide at and beneath the seafloor? So when I say microbes, I'm talking about single-cell bacteria and archaea, but also more broadly, viruses, fungi, and single-cell eukaryotes. I know we have a diverse audience. I'm not talking about octopus in terms of their ecosystem service, just to be clear. So to ask this question, Beth and I got together a group of scientists about a year and change ago. We partnered up with DEUCE, the Deep Ocean Observing System, and we brought this group of scientists together to sort of come up with a list of essential ecosystem services that microbes provide in hard rock crustal environments in the deep sea. And this was really done in the context of deep sea mining and thinking about how would these ecosystem services be impacted by mining and how can we identify thresholds of harm to those services. And you'll recognize Gus, who you also just saw, Miguel, and of course Beth. And so you'll be hearing from Gus and Miguel after, and all these people contributed to the work that I'm showing today. So we came up with these 10 ecosystem services that are provided by microbes in the deep ocean. 4 of these are supporting, 5 regulating, and 1 provisioning. And we're working on the manuscript for this now, building off of a paper that we published about 4 years ago now. But what I want to do today is talk about these services in the context of the Octo Odyssey expeditions and how we're trying to address them, in particular at these sites of fluid discharge. And so this is something we collected an incredible sample set to do, but we didn't really realize it until we were all out on the ship together that we had the opportunity to do this. So over the summer, Gus and I wrote a proposal to try to get some funding to develop this work. And we weren't successful, but we still have this beautiful infographic that Gus created, yes. And we'll continue to try. In my lab at Hui, we focus on these warm-venting fluids coming out of the seafloor and working all around the globe and the microbes associated with it. And you'll be hearing from Gus in just a few minutes about the rock microbiome, both the colonization experiments he put on the seafloor, as well as the natural rock community. And the dream is to then connect it to the octopus microbiome. So we collected this incredible set of samples with all of you that are sitting preciously waiting for the money to arrive, but to really kind of look at this whole connection from the subsea floor fluids up into the rocks and whether or not they're influencing the animal communities we find there. So these are some of the questions that I'm addressing in the crustal fluid microbiome. So what biomass is available as a food source? How are microbes contributing to carbon and nutrient cycling? And I won't answer these questions. Do microbes help or hurt the octopus? Which again was one of the driving questions that funded this work originally. And what is the scope of genetic diversity here, which both Gus and Miguel will be addressing. So we did a lot of stuff out on the ship, collecting all the samples to do a lot of work, much of which has not been done in my lab yet. But I'm going to focus on the first two, which is really quantifying the biomass and detecting activity. And the first ecosystem service I wanted to talk about is basically microbes as a food source. Like lots of things eat microbes in the deep ocean, and the simplest measurement of that service is simply counting how many cells are there. But before I get into it, I wanted to remind you that all the fluids at these 3 sites of discharge are not identical. So this is data from Jeff Wheat, who was on the December cruise, and you'll recall we had temperature loggers that we left for the 6-month period in a lot of the sites that had discharge. And then when we went there, we put a dissolved oxygen sensor in the venting fluids as well. And so this is a plot showing dissolved oxygen on the y-axis and temperature in the x-axis. And you can see that the oxygen and temperature patterns are different across these 3 sites, which really reflect probably how long the fluid has been in the crust reacting with rock. And this is kind of the telltale of these ridge flank crustal fluids. In every place we've studied them, which is like 3 places, this being the third, is that you have this decrease in oxygen as temperature increases. And to be honest, we don't really know why oxygen goes down. It could be an abiotic reaction, but it could also be microbial respiration. So this could also be an ecosystem service. We just aren't sure of that yet. So we also collected these fluids and we just counted how many microbes were there. So up on the y-axis now we have the cell concentration across the 3 sites. And what's really important is that microbial biomass in this environment is not enriched compared to surrounding background seawater. And so that gray out in the top there is kind of background seawater concentrations from each of the 3 sites we visited. And this is really in contrast to the work that we also do in our lab at mid-ocean ridges, right, where we have these big, big chemosynthetic ecosystems and where we have an order of magnitude or more cells. And I think it's important to remember because despite the abundance of life we see at these seamounts, at these outcrops in Costa Rica, this is such a different system. And I constantly have to remind myself of that as well. So we have a standing stock of biomass that is much like the rest of the deep ocean, and it's really quite low. The other thing I wanted to say about these fluids is, Rachel explained so well how this happens, and there's also this really strong tidal signal in these fluids. This is also data that we are able to extrapolate the signal from based on that thermistor data. So now on the y-axis is about 18 hours of deployment during our December cruise. This is work from Andy Fisher and his student Rue, who were out with us in December. And basically this is the temperature, the actual temperature data in the open circles up and down. And then this is the model data with the tidal surface cycle. And so we have this diurnal lunar cycle within the data. And I was trying to think about how to explain this, knowing that Rachel was going to talk in front of me. And I remembered this drawing that Carlos did, right? The hydrothermal siphon's a little bit hard to understand, right? But I really love this drawing. And you can think about the physics-driven part of it, right? But you can think about the tides kind of like the dragon, right? That they're also pushing and pulling on Earth's crust. And I think this is really important to understand because at some point in time, we actually saw some of the vents shut down on our cruise. So we were at, I think this is marker A, the final dive of our cruise. Jeff Wheat was measuring temperature and dissolved oxygen, and then I jumped in there with my fluid sampler, and the vent just disappeared right before our eyes. And when we plotted that against the tidal cycle, we were at one of these lows. So the dragon
couldn't blow hard enough, right, to overcome. There wasn't enough pressure-driven circulation to keep that hydrothermal siphon going. And other sites don't appear to be like this. Is marker A, I think. I don't totally remember because I was sad and I left the van. But I think it's important, right? So if these microbes coming out of the venting fluids are an important food source or providing some other service, this isn't a steady state flux as you might have in some other systems. So the other ecosystem service I wanted to mention is the synthesis of new organic carbon, which is really driven by microbes that can fix inorganic carbon into organic carbon. This is usually driven by chemical energy, and again, deep sea hydrothermal vents are a great example of this chemosynthetic reaction. And of course, the question is this sort of thing happening in these octopus nurseries? And I will say in the one other ridge flank system that I've studied really well, we do see evidence of this even without the presence of charismatic megafauna. So those of you who are at sea with me in December know that I set up these really laborious experiments where I basically took the fluid coming out of these seamounts and I incubated them with either labeled acetate or labeled bicarbonate and set them up just sitting at room temperature for over 4 days, and then did a couple of things. And the one thing I wanted to show you today was we've just done cell counts on all of those experiments. And up here is marker W at Dorado, and down here is marker Q at La Pulperia. And basically what we see is that in all of these experiments, especially the acetate and bicarbonate conditions, microbial growth is indicated by an increase in cell concentration compared to an unamended control. But microbes are also really good at growing in bottles. So this could be a little bit of a bottle effect at the end. And so what I'm doing right now is sending all the filters from these first 2 time points to a facility to look at the uptake rate of that isotope into the particular organic carbon. And then we can actually find out who is taking it up versus just growing well in a bottle. And then I also sent samples from the cruise to Sunisha Walter, who's at the University of Delaware. And she is measuring the dissolved organic carbon in these fluids. So Rachel mentioned DOC. The 2 other systems where we've studied DOC, ridge flanks are actually a sink for dissolved organic carbon, and that's actually really important for the global organic carbon budget in the deep ocean versus a mid-ocean ridge, which is a source. So again, if we want to try to understand carbon cycling in the deep sea, we have to get these numbers. So I just wanted to show some preliminary data, remind you that these fluids they're really unique in the ocean in terms of being depleted in oxygen. They're also quite low biomass. That does make some of our methods a little bit more challenging. And this tidal signature in the fluid flow is just kind of a cool thing that we found. We've seen it in some other deep sea environments, but in this case, I've never seen a hydrothermal vent shut off in front of my eyes. And it felt precarious to me, right? There were a couple octopus in that crack, and they went away when the fluid stopped. They hadn't laid their eggs there yet. We have this low biomass, and our microbes appear to be stimulated by both an inorganic and organic carbon input. And just a reminder, we have big dreams, big hopes, and like I said, we have all these octopus samples, and Gus and I spent a lot of time going back and forth with you all to say, okay, what exactly is here? Because not only do we have octopus tissue samples, we also have some eggs, we have some other cement. I mean, we're really set up to look at this connection between the microbiome of these different deep sea substrates and what's happening with this remarkable, remarkable animal. So I think that Gus and Miguel are going to address some of these other ecosystem services. So Gus is coming up next to talk about the rocks and a lot of it with some really great genetic data. And finally, I just wanted to thank everyone who brought me onto this expedition. I think Beth said this, but I have been going to sea for a long time, and it was really just kind of a remarkable cruise for me. Felt like I was quite jaded before this cruise, and I think my favorite part was just watching all of you tell the story of your ocean. It was just such a wonderful thing to witness, and I'm really appreciative to our funding as well. So that's it. Thank you. Do you want me to introduce Gus?
Okay, so we have time for questions.
in the ecosystem service in providing, you only have genetic resources, but do you think there's some connection between the deep and the shallow waters for the fish and food and that connection with the fisheries? No, I'm meaning connection of the deep with the shallow, or because it gives the impression that they're completely disconnected. So there's something going up here, something like that. But could there be connections of the deep with the shallow? So that would be another service. Or providing support to other animals.
Yeah, I think in the first one, supporting, when we were talking about a supporting service, we were thinking about symbiotic relationships, not necessarily obligate either. So you could imagine in like a gut microbiome of some migratory species or something like that as well. And I will make sure that, because we're writing the paper right now, that we make sure that's in there. Any other questions?
Hola. Thanks for the presentation. It was very interesting. I have a question with the vent showing down and shutting down. Is that connected with the counterflow that Rachel was talking earlier, or is this because of something else, geologically or physically?
So the shutdown we saw was at Dorado, right? And so Rachel should probably answer this question. This was a part of Dorado that had very weak venting in the first place, and my guess is just the pathway to that site, like if you think about highways through oceanic crust, that the push just isn't hard enough to overcome that tidal signal.
Yeah, so we actually left. We actually left thermistors between 2013 and 2014 in 6 different locations, and 3 of them intermittently shut down and then turned on again. So it is, tidal forcing just means that it's trying to push it back in when it's like weakly coming out, and it's like, and then it maybe finds another path. Because the other thing is other things turned on that were off. So, I mean, I think it's complicated, but the only reason that happens is because it's so permeable. If this were a relatively impermeable system, tidal forcing would not have that huge an influence on it, and it's a function of how easily fluids move in and out of the crust.
Yeah, the other connected system that Rachel and I both worked at, the seamounts are heavily sedimented except for the very, very, very tippy top. And it's much warmer. We haven't seen that type of shutdown. Oh, and what I meant to say is if we ever go back, we should use tidal forecasting to decide when to do the fluid sampling.
Thank you so much, Julie. So, thank you again. So, the next presentation is Gustavo Ramirez. This one is, yes. So, welcome, Gus.
Thank you. I wonder if we're doing a good job following Carlos's performance. I feel like Paul McCartney showed up and played, and then I'm supposed to say something? It's like, anyway, my name is Gustavo Ramirez. I'm assistant professor at Cal State LA, University of Los Angeles, and I love microbes. I especially love to try and understand how microbes interact with the world. Okay, and the world can be abiotic substrates that are all around them, or biological substrates that are around them. So anyway, today I will talk about the microbial ecology of seafloor exposed crust in La Pampa Submarina here in Costa Rica. I always start my presentations with a picture of the Earth system. So again, I said I like microbes interacting with rocks, right? This is a pretty big rock, okay, and it's full of microbes that interact with it, and it's submerged in an ocean. Okay, I'd like to point out though, now that I'm perfectly set up, I don't have to cover a lot of my background. This is not the only wet rock in the vicinity, right? Around this star that we live, there's plenty of other wet rocks where a lot of the activities, biological activities that we'll be talking about today, could also be happening. So I'm an astrobiologist at heart. So anyway, pretty cool rock here, tons of microbes in this rock. All right, what's special about this rock, however, is that when you whack it open, it's got a very hot core, okay? So that heat consequently has to go somewhere, right? That's thermodynamics, heat flows. Luckily for me, Rachel explained a bunch of this already. So this is a picture, or this is a map of the world that shows the net surface heat loss and where that happens in the world, okay? It's important to point out that here in the eastern equatorial Pacific, there's a ton of heat loss that's expected, and that's the anomaly that we see there. Even more importantly is the fact that heat is best transferred directly from the top layer of the lithosphere into water, okay? And places where that happens is actually places that we call sea mounds, or I guess if they're tall enough, we officially call them sea mounts with the T, right? And sea mounts are, again, just layers of the lithosphere that we've been talking about here for the last couple of talks that are, the lithosphere is uneven, of course, and some of those places, the lithosphere becomes or comes into direct contact with the hydrosphere there. So it's just protrusions of the crust above the overlying blanket of sediments that usually covers the abyssal plains of the ocean. So again, the uneven heating across this top layer of the lithosphere ultimately fuels this hydrothermal siphoning that we've been talking about here for a while, that brings in what's ultimately bottom seawater from really deep bottom seawater here with a very specific thermal, geochemical, and microbiological signature. It brings it in to this sub-sea floor crust, and then it goes into this flow path. I know nothing about how to predict the flow paths, but at least one modeler is here that can expand on that if people are interested on that. But what I really care about is the following. As this deep sea water comes in, right, it evolves. And that evolution is happening physically, right? It's changing temperatures. It's evolving geochemically, right? It's changing the geochemical signature of the water. And most importantly to me, it is evolving microbiologically, right? So the microbial communities that eventually are found in the water that's venting at the warm spots of focused venting, for example, El Dorado Hill here, are fundamentally different in structure and composition to the microbial communities of the background seawater, right? And again, all of that change happens in this flow path. Okay, what else I want to say about this? Yeah. This ultimately geophysical property, right, of water flowing into and out of the seafloor crust has immense ecological repercussions. Okay, so what we see here is just the differences in the animals that are seen at different places where water either enters a seamount or it exits from the crust at a nearby outcrop. And again, this is the Dorado Tengolset system. You'll keep seeing this system over and over again because as what we've mentioned before, this system is probably the best studied proxy for this phenomenon on Earth by now. So anyway, what you see here is the surface of either Tengolset or Fuente. One of the big seamounts here that we know is where the warm water, the cold water goes in. You see a crazy amount of very diverse animal life, sea sponges, anemones, corals, right? And what you see here was Carlos painting this remarkable painting of the kind of life and the activities of life that we observe where the warm water is coming out, right? So this is where water's going in, and this is the animal activity that's really the dominant showpiece of what's happening with biology, where the water's coming out, right? So there we get the warm water basically attracting hundreds, if not thousands, of octopus, deep sea octopus females, right? That we were able to explore in our last two expeditions to La Pampa Submarina here. And again, this is Adoración al Dorado? Maternidad Adorada is the name of this piece right here, so I had to include it. Anyway, so a lot of ecological repercussions, right, to what's ultimately a geophysical phenomenon, right? That's the point that I'm trying to make here. One thing that's absolutely missing from both of these depictions of the ecology of seamounts is the following. The thing that's missing is that in every cubic centimeter of water that you see here, there's approximately 1 million prokaryotic cells. Okay, so that's bacteria and archaea, and I guess fungi, right? A lot of microbes are playing a lot of important roles. And those roles are also affected by the hydrothermal siphoning of the system, and those roles, right, biogeochemical cycling, nutrient generation, right, may also affect the macroecology that we're able to see with our own eyes with the large animals. So again, the role of microbes and the ecological repercussions of microbes being ubiquitous across these ecosystems is often ignored. I guess that was the case because of where the technology was at the time, right? But we're at the point now where through next generation sequencing and high performance computing clustering analysis, we're able to incorporate microbial activity, or hope to incorporate microbial activity into this broader regional ecological activities, okay, fueled by the hydrogeology of the ocean. Anyway, so we've seen this one before. The dream, as Julie correctly pointed out, is sequencing, generating microbiome libraries, metatranscriptomic libraries, metagenomes, as well as single cell sorting, right, of this incredible suite of samples that we were able to collect last year during the Octopus Odyssey expeditions. Again, we've got hundreds and hundreds of tissue samples that were masterfully dissected by Fiorella and by Holly. We've got tons of rocks with a lot of metadata on each one of those rocks, tons of fluids and tons of background seawater. Again, the idea is to really understand the microbial ecology in terms of a sort of a primary succession directly on the abiotic and biological substrates that are present where the warm waters vent. So that would be the rocks directly where there's warm venting happening. And then also the animals that live right at the site of venting warm fluids. So that's really the dream. Does the microbiome of the venting fluids help the mothers or the octopus females carry on with their brooding cycle that can last up to 4 years, according to deep sea octopus experts? Does that microbiome maybe generate specific biogeochemical cycling of the rock that allows the embryos or the mothers to maybe have disproportionately higher access to reduced nitrogen or phosphorus or sulfur, another key nutrient, right? Those are the big ideas that we're going after. So anyway, yeah, this is still TBD. We're waiting for the big agencies to take the bait. We're almost there. And what I'll do for the rest of the talk, however, is focus on talking about the rock microbiome from a subset of samples that were collected in this expedition. Specifically, I'll be talking about the rock microbiome explored, or the ecology of the rock microbiome explored through field ecology experiments that my laboratory put together and that we deployed at La Pampa Submarina for a total of 6 months. So, yeah, like I mentioned, we've got hundreds and hundreds of samples that were collected during this 2 legs of the Octopus Odyssey expedition. What my laboratory's done already is extract DNA and sequence the microbiome of 107 samples already. Okay, so we try to get an even spread of what the samples are. The majority of the samples are comprised of sediments, comprised of natural rocks, and then I've also gone ahead and sequenced all of my experiments, incubation experiments. Now, I'll talk about that in just a second. So what you see here is a 2-dimensional plot that shows different circles, okay? Each one of those circles represents the microbial community structure of a single sample's microbiome, okay? If 2 circles are very close to each other in this 2D summary of something called the principal coordinate analysis plot, if 2 circles are close to each other, that means that their microbial communities are extremely similar, okay? If 2 circles are further away from each other, that means that the microbial communities are actually much more different across those, or in those 2 samples, okay? So that's the representation that I'm trying to show here. What you see here is that there's clustering based on these colors, right? And what these colors show is what these, what these colors show is the substrate type that each one of those samples comes from. So there's more similarity between all of these all of these green dots here, which makes sense because we would expect that microbial communities sequenced from individual natural rocks that look like this, right, would have microbial community compositions that are most similar to each other. That's what we're seeing there. Same thing with this cluster representing deep sea sediments collected with push cores. And then this other cluster here represents my microbial incubation experiments. And what I'll do for the next couple of slides is focus on the data that's extractable from this cluster here that's in purple. These are microbial incubation experiments. So let me go ahead and talk about that just for a second here. So as a microbial ecologist, I am fundamentally interested in understanding what environmental factors drive microbial community structuring, okay? And then a secondary question from that would be microbial community activity, okay? But for now, we'll just talk about microbial community structuring. In order to understand what environmental factors drive the structuring of microbial communities on rocks at the seafloor, we set up this deployment experiment. So each one of our experiments was basically comprised of 50 grams of sterilized substrate that we think had the potential to tell us an interesting story about the ecology of the Pampa Submarina microbial ecology of La Pampa Submarina rocks. Okay, so again, one of these experiments is comprised of these 5 substrates individually housed and deployed in dive weights using the Sebastian ROV, of course. 2 of these 5 substrates were actually basalts. One of them were considered fresh basalts from the western part of the East Pacific Rise, only 20 years old after the volcanic explosion when these were collected. And then we also have native basalts that, as we heard before, represent 23 million-year-old rocks in this area. Okay, thank you. Yeah, we had mild steel, which is a synthetic metal that's known to leach a lot of iron when interacting with seawater. We had a glass wool that is basically inert, just borosilicate shavings that do not interact with water at all. And then I had a manganese carbonate called rhodochrosite. And again, one of these is a single experiment. Okay. Hope I can get through this in a minute and a half. So anyway, long story short, these experiments were deployed at 5 different sites in the seafloor. Okay, 3 of the sites were at El Dorado, and one of those sites was at La Pulperia, with the 5th site being at a site where there was absolutely no venting that was visible. So hydrothermal venting was present for 4 of these deployments, again, 3 at Dorado, and 1 at La Pulperia, no hydrothermal venting for the 5th deployment there. You can see by the presence of the brooding females there that 4 of these sites, 3 Dorado, 1 Pulperia, had venting again, and then this was our environmental control where there was no venting at all. Long story short, there's a lot of questions that can be set up to be answered by a data set, by the data set that we collected from these experiments. A very simple depiction is shown here. Okay, so what I have here is only substrates that are either 20 years old, thank you, or 23 million years old. 20-year-old substrates are in golden color here. 23 million-year-old substrates are in gold color here, and 20-year-old substrates are in red color here. And with this data set alone, we can ask extremely simple questions using something called an analysis of variance, such as this one. Does basalt age affect the microbial community in incubated natural substrates? In this experiment, the resounding answer is yes. Does location affect the microbial communities in incubated natural substrates? Again, in this experiment, the resounding answer is yes. Then we can even come up with multiple predictor models that allow us to ask a more complicated question as to which one of these environmental variables affects the communities the most. And of course, I always wanted to ask whether or not venting affects microbial community composition, and of course it does. So anyway, this is a super simple experiment, just a lot of preliminary data. I won't have time to get into all of this. And I'll close by saying that, okay? Oh, okay, yeah. I'll close by saying that these are the first baby steps answering truly like first order ecological questions about what environmental factors dictate what the microbial communities do and how they assemble in seamounts of La Pampa Submarina, Costa Rica. And again, the goal is to be able to get the money to integrate these observations from the rock with all of the other substrates that are patiently waiting for us to extract DNA from them and sequence them. And with that, I'd like to thank the funding agency. It's Schmidt Ocean Institute. And just to name a few here, a lot of this work came out of my graduate student's thesis, Oscar Almacen. Thank Don Jorge, thank Beth. Thank you all. Take it.
Thank you, Gus. We have time for one quick question. No. Okay. Thank you. Okay, we will continue with the presentation of Miguel Semedo, and he sent a video, so they will be online. So Miguel, he received his PhD in marine sciences. Now he's currently a junior researcher at the Interdisciplinary Center of Marine and Environmental Research in University of Porto. As a microbial ecologist, Miguel is interested in investigating the diversity and function of microbial communities across multiple environments and their roles in biochemical processes. So,
I'm sure you'll have a great time, and hi everyone, and good afternoon in Costa Rica. I'm sorry I wasn't able to join in person. I'm a little bit jealous of not being there, but I'm sure you'll have a great time. And I also want to thank the OSCRI organizing committee for accepting this talk in their program. And yeah, I just hope that you have a wonderful symposium and enjoy the time there. So I'll be talking about metal impacts on deep sea microbial communities and natural recycling potential. And this is an ongoing research, but I will present a few observations from the Octopus Odyssey expedition that we joined last year there in the Pacific Ocean in Costa Rican waters, and it was such a privilege to be there. So I come from CIMAR, a research center in Portugal about marine and environmental research for the University of Porto. And so here, the research that I'm going to present today, it's also part of an ongoing effort of trying to understand and monitor potential impacts from deep sea mining operations of earth minerals such as cobalt, copper, lithium, nickel. So this is basically an emerging economic activity due mostly to the decline in mineral land stocks, also an increase in global demand for the energy transition, and also due to the fact that the mining the deep ocean is now less challenging technically compared to a few decades ago. But of course, this is also an ongoing debate. We know very little about the deep ocean. We still know very little about what seabed disturbance what harm can cause seabed disturbance to the deep sea benthic ecosystems. So there are a few exploration contracts in international waters, shown here in this figure in green. But the full-scale commercial exploitation is still pending environmental assessments and regulations, especially in international waters. And these assessments and regulation also are very important today because we know that the baseline for our knowledge in the deep ocean compared to other ecosystems is relatively poor, and we also don't know very much what are the thresholds of harm in these environments. So how much harm can they tolerate, and how slow will they recover? Some countries, of course, this is mostly I was talking about international waters, but some countries are also exploring the possibility of performing this activity in their own national waters, such as Norway, for instance. But a lot of other countries have a more precautionary approach, since we don't know very much what can these activities do to the ecosystem. They are still they won't advance with regulations and with full-scale implementation before better assessments are performed, such as, for instance, Costa Rica, I think, as a precautionary pause agreement, and also Portugal, my own country, until 2050. So, and the main concerns related to the environmental impacts of deep sea mining are sediment structure modification, sediment resuspension, and also habitat removal, the removal of unique and special habitats, the potential release of toxic concentrations of metals to the surrounding seawater, and that's the impact that I'm particularly looking at, and also physical-chemical changes, say changes in the pH, in dissolved oxygen, and so on and so forth. And all these consequences can have impacts on marine life, and including microbial communities. Microbial communities are known to perform multiple ecosystem service functions and services that are very important for humankind. They are a huge reservoir of genetic biodiversity. They also contribute to regulate greenhouse gases production, such as methane or nitrous oxide. They are important for nutrient cycling, and I will be talking a little bit about nutrient cycling today. And they are oftentimes the basis of the food web in these ecosystems. Regarding the two microbial ecosystem services that I will be talking about, biodiversity and nutrient cycling. Nutrient cycling, as you know, is basically the cycling of essential nutrients in the ecosystem, and I'll be focusing a little bit on two metabolic pathways of the nitrogen cycle. Nitrification, shown here on the top in the right, which is basically the oxidation all the way from ammonium to nitrate, and this is basically a chemolytic autotrophic process. Uses oxygen, but this is a very important nitrogen pathway. And the nitrate that can come from nitrification and also from other pathways and from ocean circulation, that nitrate can be further reduced to nitrous oxide and the nitrogen gas through denitrification, shown here at the bottom, which is basically a major heterotrophic pathway, a respiratory pathway that generates energy mostly in anoxic conditions. So these two pathways are very important, but we know very little about what are the metal impacts on deep sea microbial diversity and crucial pathways such as nitrification and denitrification. So thus, our investigation tries to investigate potential effects of cadmium. This is what I'll be talking about today, but other metals as well that we are performing in other expeditions on microbial diversity and nitrogen cycling potential, and trying to help answering the question what metal levels can impact microbial diversity and nitrogen cycling potential and how. So as I mentioned at the beginning, we were lucky enough to participate in the Octopus Odyssey expedition last year aboard the Falker 2 from the Schmidt Ocean Institute, shown here. And we, as some of you may know already, like we visit a couple of different seamounts, and aboard this expedition, we performed some metal exposure experiments with sediments collected in these different seamounts. And basically we incubated the sediments, surface sediments, with bottom seawater at 5 different levels of metal exposure. A control without any metal, without any dissolved cadmium, and then the 4 different orders of magnitude from nanomolar to 15 micromolar. And this nanomolar level, it's a level that you find in natural conditions, and this 15 micromolar level is an elevated level that you could probably find after seabed disturbance. And as I mentioned, we visit different seamounts. The results that I'll be talking about today are only from one, Dourado outcrop, shown here, and each of these treatments has 6 replicates from this seamount. And so we then incubated these sediments under these conditions for 96 hours at 4 degrees in the dark to try to approximate to the conditions in the deep where the samples were collected in the deep ocean. And after these 96 hours, we store the sediments at minus 80 degrees for further DNA and RNA extraction. And then we sequenced the 16S RNA gene, which is basically a universal marker for prokaryotic taxonomy, to assess its composition and diversity. We also did some abundance and expression studies of some naturally cycling genes through qPCR at the DNA and RNA level, and hopefully if we are lucky enough to obtain more funding to perform shotgun metagenomics and metatranscriptomics. So a really brief overview of the preliminary results observed so far. In terms of species richness shown here on the left, and so you can see the y-axis, the observed ASVs is basically the number of observed Amplicon sequence variants, which is a close proxy to species in the prokaryotic world and in this type of technologies. And you see the different treatments here, right? Control on the left, and then all the way to cadmium 15 micromolar on the right here in red. And on the left is species richness, the number of different prokaryotic species, and on the right alpha diversity, which is that number basically balanced by the differential abundance between the different species. So, but in general, the main take home here is that besides an apparently lower species richness here on the highest concentration of cadmium, there were no major effects on prokaryotic species richness or diversity, at least at this level. So we are talking about DNA level. So no major substantial impact here from this particular method. When we look at the community structure and beta diversity, which is basically the dissimilarity across the different samples, and shown here on the left is a PCOA plot where each of these circles is one sample colored by the different treatments, and if the samples cluster close to each other, they are basically very similar to each other, but if they cluster further apart from each other, is because they are different or dissimilar from each other. And what you can see here is that there was no clear treatment effect on the dissimilarity across the different samples. However, if you also look at their distribution within each treatment, so within control, within cadmium 0.015 or within cadmium 15, if you see the variance for each treatment, it looks apparently different. And if you look at the plot, the box plot on the right, which is plotting the distances from each sample to the centroid of these circles, basically the variance or the difference between each sample and the mean positioning of these of each group, you see that this dissimilarity within each group is much lower when it's significantly lower in the highest cadmium concentration, which suggests that there is a loss of heterogeneity in this treatment that we need to further explore. When we look at nitrogen cycling genera, and of course, when we say nitrogen cycling genera, I'm just talking about sequencing data with the 16S RNA gene, a marker for prokaryotic diversity. So these are only genera that are known to potentially to perform some of these reactions, not necessarily that they are performing there at the moment. So the nitrification here, as I mentioned at the beginning, a respiratory pathway, stepwise reduction of nitrate all the way to N2 and N2. I'm showing here the relative abundance of stenotrophomonas, which is considered to be an important denitrifier and found to be very important in some deep sea sediment layers, and actually in deeper sediment layers like below 50 to 100 centimeters. And what we observed here, despite in some samples was relatively abundant, these genus, and in the highest concentration of cadmium completely vanishes. When we look at Ralston, another important denitrifier, especially across the different sediment layers from the surface to like 100 centimeters below surface, you see that even though it was not such an abundant genus, but still quite abundant, and also its relative abundance completely decreased at the highest cadmium concentrations. So some measured denitrifiers had significantly lower abundances only at the highest concentration of cadmium. When we look at another group of nitrogen cycling organisms, the nitrifiers, or in this particular case ammonium oxidizers, responsible for the initial steps of nitrification, we didn't see any major differences regarding the cadmium treatment effects. So just to underscore these initial conclusions from this study with these sediment samples, we observed that exposure to high concentration of cadmium may induce loss of heterogeneity in deep sea microbial communities. Now the question is, are we losing or switching unique taxa or unique functions from these samples? That's something that we need to further investigate with metagenomics. And also, we observed that some important nitrogen cycling taxa were substantially impacted at high levels of cadmium exposure. It seems that mostly anaerobic and dissimilatory processes may be more susceptible than chemotrophs, but also something that we need to further investigate. So now we have already performed 2 or 3 more expeditions in different locations, and we have performed these repeated experiments with other metals as well, copper, for instance. And now we will also plan to advance with transcriptomics and metagenomics, of course. With that, I'd like to thank you for your attention and thank a lot of all the CIMAR researchers that helped with this research, also external collaborators and the funding sources, obviously, for the support to participate in this expedition. And last but not least, all the amazing scientists that I was lucky enough to meet during the Octopus Odyssey expedition. And I just want to, I hope some of you are there together and enjoy a little bit of the science of this symposium and some free time as well in Costa Rica. Bye-bye. Thank you.
Well, thank you to Miguel. So we don't have time for questions, but we are going directly to the coffee break. Please make sure the coffee break because we are on delay. So we have 15 minutes for the coffee break and then coming back. Please. afternoon. Good afternoon, everyone. ¿Sí? Voy a empezar a llamar ya. Voy a empezar a llamar ya. No, creo que está llamando a la gente. Sí, va tardísimo. Comenzamos. Good afternoon, everyone. Welcome to the last session of today. We will start with our first presentation with Keylor Rojas Jimenez. He's a professor at the Department of Genetics and Biotechnology, School of Biology of the University of Costa Rica. For more than 25 years, he has conducted research in areas including biodiversity, genetics, ecology, and biotechnology of microorganisms. Where is Keylor? He's in here. The floor is yours.
Many thanks. Good afternoon. I was deeply moved by Carlos' painting, and I think that artists and philosophers will save us in the middle of this jungle that we are now. And so that's why I'm going to change my talk, at least how I move, and I would like to make it more philosophical. So what I would like to propose you, and I hope to convince you, is to change. I have this question: what is life? Or how do we define life? Let's say, so we can take different perspectives, but there is different ways on how we can determine that something is alive. And so I hope to sell this idea using some data from the deep sea. But also, since we had this idea, we had the idea, but we didn't have data. So we get public data, and we hope that in the future we can work with our own data from the expeditions we have. I'm just going to mention this concept of plasmidome. A plasmid is a genetic element that is, we have, for example, a bacteria. It has a chromosome, and it has some regularly circular DNA close to it, that it has its own particularities that I will try to convince you that they are alive from a new perspective. And so all these entities in an ecosystem are called the plasmid. So so first, just from your biology classes, just to remind you some basic concepts of mobile genetic elements. That is genetic material capable of inserting itself into the genome of another organism. So in a way, it's like we can call it cooperative or parasitic DNA. They facilitate the movement of genes within and between genomes. And then they contribute to genetic diversity evolution. And so within this, we have plasmids, transposons, insertion sequences, and integrons. So this is DNA in different forms. And so, in a way of representing this, is that, for example, we have small elements that are within others, that at the end we have plasmids, and these plasmids have different genes that can move to other species. So this, a better way to represent that is like this. We call it maruscas. And so, but that's the way they are in nature. Um, so, what we say is that you know the many people say, or for years there have been these people saying that are the viruses alive or not? At least myself, I'm fully convinced that viruses are alive, and we just experience what a virus can do in the last pandemic. And our humble proposal is that the mobile genetic elements, the plasmids among them, are in the same category. So this challenge on how we define the tree of life, so we know there are three main domains. So what new people are proposing is that there should be something above that, what they call series. And one series has cytota, everything within membranes, and acetota, not membranes. And so that's where viruses might be, and what we are proposing is that the plasmids are within them. So the current definition of what is alive, it has many, let's say, in my point of view, distorted requirements. But I think that since having evolution of how we consider life from the Greeks until now, it has to change. And a basic definition of life might be related with the capacity of propagating the genetic material by itself or by the help of others. And Don Jorge gave me one book that one can add that is restricted by the laws of physics, which are very simple. But I think that we have to challenge the concept and expand what we consider what is alive. And as a proof of concept, we wanted to examine these plasmids in a very unique and fragile environment, as is the deep waters, and see how these new entities or biological entities behave. And so we wanted, since we had this idea, to propose a case study using the global deep sea plasmidome. Since we had no money and no sequences, what we used was the data from Malaspina and Tara Ocean expeditions that they did deep research. However, if you read their papers, there is no mention of plasmids, and so we are adding this, let's say, new component. And also because we know very few about marine plasmids. In databases, they represent less than 1%, although there might be more, you know. But we have this bias for studying, I don't know, clinical microorganisms. And then we use these 81 metagenomes from Pacific, Atlantic, Ocean, and Indian Oceans, from two depths, from the mesopelagic layer and then the bathypelagic layer. So we wanted to see how this community of new entities are there. And beyond this, if it is a new entity, one can start talking about the taxonomy of plasmids, that something that is not well defined. Or furthermore, we can talk about plasmid ecology or even evolution of plasmids, and something that we are doing currently in my lab. And for this, we developed, together with some colleagues in the University of Oslo, a pipeline of bioinformatic pipelines in order to extract all the information possible from metagenomes, trying to obtain information of plasmids, but also we have another paper on the virome, for example, also from the deep sea, and we are also working in other environments. And with this is in GitHub, it's called Metagenomics, and so it's available for, we can also get information about fungi or also MAGs, metagenome assembled genomes and other things. So try to get the large amount of information if it is possible. Okay. And so here are some results just to show when we analyze the composition of who's there. So I'm going to go very deep, just you see that there are different colors. And this is up here is when you analyze just the let's say, the host, the parent cell. And then here you analyze, when we analyze the plasmids. And so these plasmids are assigned to different groups. And so just a simple thing is that there is a different thing. They are different one from another. In this case, most of plasmids belong to alpha proteobacteria, gamma proteobacteria, and actinobacteria. Let's say from different groups, but the thing is that the global population of the host is different from the global population of these entities. They have, in this system is more or less similar, but we have studied, for example, here in polluted rivers, and it's totally different. They behave differently. They have their own sense of meaning of population. And then when we analyze specifically, for example, the plasmids by different oceans or by layers, the mesopelagic has almost double the amount of species. So there is a strong influence of depth on the amount, on the richness, but also on we use another alpha diversity indices. And so there is a strong effect of depth. But also we saw that Atlantic has a higher amount, a little bit more. We still don't know why. When we do the analysis that Gustavo explained very well, just to see that the structure of the communities of plasmids, they cluster together by depth. So these are the ones of the bathypelagic, these are the ones of mesopelagic. And then, so this is a map of the global deep sea plasmidome. Actually, this paper is, we have to respond by tomorrow, the reviews. But we have to, I think that we'll have it accepted in a couple of weeks. But what we see is that this is all the places that we analyzed and see the sizes and also the colors. And so what's other thing that is that we found more plasmids in the mesopelagic layers than in the deep layers. But also we looked for antibiotic resistant genes, and surprisingly we found very few, very few, actually only 7 in the whole databases that we are talking about, millions of sequences. It gives a little bit of hope that deep sea is not, or still not, very polluted. But let's say, because for example here in Virilla, one river that is close here, we found like 40 different antibiotic resistant genes in one river. But here, not so many, and so far in the superficial layers. And here another is we look for different metabolic pathways, and I'm not going to go in depth, but we see that there are the plasmids are providing the ecosystems or the host in that ecosystem different characteristics there, and they have some variation in different sites. But one of the most interesting is in the Atlantic, that they also have these genes related with hydrocarbon degradation, which is a little bit interesting. And so I don't know if it's more related with pollution or not, but it's something to analyze further. And also we analyze the different mechanisms of mobilities, how they change or can transfer genetic material from one to another. So there are differences the strategies they are using to transfer genetic material from one to another. So in summary, we found differences between depths, and then in the upper layer, larger diversity, and also, let's say, antibiotic resistant genes, but still very low. Yes, I think that's my message. Thank you very much.
Okay, due to the delay that we have only time for one quick question.
Thank you. Sorry, Gus, for taking your question. Thanks, Caleb, for the presentation. I think your goal was achieved of seeing things differently. My question is if you think that maybe the high richness in the mesopelagic community, let's say, is because of the different oxygen ecotones. And looking at the functional annotation, it catches my attention that there is no shared, for each of the basins, there is no shared gene for the three of them in that table you showed. So I was just curious about that. Why do you think that there is no specific functional gene for the three basins?
Actually, this was the old figure they asked me to change to a different color scale, but there are some few functions shared, but yeah, we haven't explored in depth why are different, but I don't know. Oxygen is a good idea. We are looking at their databases for that, but we haven't found for all the samples, so we don't have that. But certainly, depth is not depth per se. Depth is a lot of environmental variables. Let's say hydrostatic pressure, or I don't know, oxygen, maybe temperature, other variables that let's say there are. I would say under the big umbrella of simply depth that we had, but certainly we have to explore more for other environmental variables, more specific. Yeah. Thank you.
Thank you very much. Okay. Our next presentation is in charge of Yvonne Rodriguez Ramirez. She's a biologist who recently completed her studies in molecular biology and biotechnology at the University of Costa Rica. Her research focuses on the characterization of marine fungi from deep sea sediments in Costa Rica and their biotechnological potential.
¿Esto cómo funciona? ¿El grande es para? Okay. Hi everyone. So we're already in the finish line. So thank you for being present, being here. I know we're very tired. Everything has been very interesting. I want to make a quick disclaimer. When I speak in English, my brain, it's a little bit slower, so I'm going to be stuck in some places, but just bear with me. And also this presentation that is here. I based this presentation in the one that I used for presenting my thesis. This is my undergraduate thesis. So there's just my name here, but I didn't do this alone. Caleb, which is my mentor, he had like a big part of it, and also Don Jorge was part of my committee. So yeah, I'm gonna talk about fungi, and I wanted to change the name to something more dramatic, like the forgotten side, dark side of deep sea microbiology, because I do believe that fungi are this kind of like forgotten side of biology. I see it like in microbiology there's like 3 children in marine microbiology. The oldest sibling is Archaea, of course. The youngest one that gets all the praises is bacteria, and then the middle child that doesn't get a lot of attention is fungi. And I'm a middle child, so I can feel it. So I wanted to start by this diagram. of what fungi do in our planet. So we know a lot about what they do, let's say, in forests or in terrestrial ecosystems, but if we go to this part, to this part of the image here, in the sediments of the sea or deep sea, we can see that there's no arrows, no diagram, it's an empty space, because we don't know what fungi in marine sediments do, and we don't know with certainty. So that's something that I really, really want to know. So here in Costa Rica, there was a paper published from Don Jorge Kaylor and other scientists about fungal communities in the marine sediments. And they actually found that there's fungi in sediments. They are present, they exist, marine fungi are a thing. And they found more than 700 amplicon sequence variants. So it's basically more than 700 species, you could say it like that. That's not exactly accurate, but it's something like that. And from that percentage, more than 50%, almost 60% of it was from fungi, distributed in these percentages. So most of them are Ascomycota. And actually, when we found in the experiments that we did for my thesis, we found the same thing, and it's also very, it aligns with literature internationally. Ascomycota are the ones that are more present in marine ecosystems in general, and also deep sea. So this was with metagenomics, and the next step from that is to actually grow the fungi. It's very different to have a sequence than to have the organism that you can see it and you can do experiments with it and actually get to know it. So why is this important? Why are fungi important? So they live in an extreme environment. We all know about deep sea, so I'm not going to explain that part. So that makes that the biotechnological potential is very high because they have, let's say, stronger enzymes or stronger metabolic tools that can be used in industry. Let's say if an enzyme can, let's say, survive higher temperatures, then it could be more useful to some industries that need more higher temperatures or changes in temperatures, something like that. But not only the biotechnological potential is important, also the ecological one. And the fact that we don't know a little bit about it, we don't know that much actually, it's something that we need to change. There's some evidence that fungi are they're partaking in some metabolism pathways, specifically from the metabolism of carbon and fatty acids. So we wanted to see which enzymatic activities they have, so we could have a general idea if the fungi from deep sea sediments actually have a role in these types of metabolism. And does that mean that they actually do it when they're in the deep sea and not just in the lab? So that's what we did. We cultivated fungi from the deep sea, which for me, that's very cool and amazing. And we did it with, of course, the help of vessels to take the samples from these three different, we took samples from these three different expeditions, from Atlantis in 2018, Falkor 2019, and then Sally Ride 2021. We have samples frozen from the more new expeditions from Falkor, but we are still not, they are waiting for us to analyze them. So what we did is we took one sample for a push core of all of these sites, and I just want you to see this one right here, because in this point, it is very near Cocos Island, we found something very interesting that I'm going to show you soon enough. So we analyzed a total of 18 samples, and what we did is that we took the sediment, we resuspended it, and then put them in petri dishes to grow and to see which fungi were there. Then we saw them. Then we wanted to know who they were, so we took DNA samples, and we wanted to know what they could do. So we made some enzymatic activities. All of these enzymatic activities were analyzed, and also surfactant activity. So this is what we found. There were many fungi in the sediments that we analyzed. And just to put this in perspective, the total volume of sediments that we analyzed was like 1.8 milliliters, because from each core we took just 0.1 milliliters of a sample. So it's actually in a very, very small volume, there were a lot of different species. So we found 55 morphospecies. We have them frozen, and that means that now the university has a marine fungal strain collection, which is something totally new that the university didn't have before. So that's great. And here are some of the little guys that we find along the way. So from these 55 morphospecies, we took DNA from 27 of them. And then we saw that all those 27 were actually 14 different strains from 8 different orders. And I'm gonna talk a little bit more specifically about one that is Ortae, right here. And that's the one that we found in the place that I was telling you to remember, that is near Cocos Island. So those 14 new strains were actually new records for the country, because before that we didn't have any record of deep sea marine fungi. And most of those strains were also, they can be also found in terrestrial ecosystems. So there's the question if they have this saprophytic activity when they're in terrestrial ecosystems, like let's say they can degrade a plant in a plantation, whatever, and they are found in deep sea and they have adaptations to live in deep sea, then are they, do they have the same activity when they are in the deep sea? Do they maintain it? So to see this, then we need to see if these strains have the enzymatic activities that they have, let's say, in terrestrial ecosystems. So this is the one that I was telling you about, Ortae. This is a black yeast, and the cool thing about this, I said yeast, and then here are filaments, but it's because this black yeast can actually grow as a filamentous fungi too. And then here are the images of the yeast. So let's go here. Okay. So about the enzymatic activities, we found that these fungi can actually degrade complex molecules of carbon, also simple molecules of carbon, and also it can degrade chitin. Chitin is in our fingernails, in our hair, but it's also in the cell wall of other fungi, but it's also in the exoskeleton of, let's say, lobsters, that we know there are in the deep sea. So these enzymatic activities tells us that they might be able to actually degrade some of the carbon that is in the deep sea naturally. So that tells us that these kind of strains have the potential to be part of the nutrient cycling in these ecosystems. But we need to make further ecological investigations to see for sure. Something very interesting about this strain was about the genome. When we first analyzed it, the species that was more like similar to the one we have was Ortae wernecki. This is very cool because this is the most allophilic fungi that there is in the whole planet. And the genome of the deep sea strain was actually bigger than the genome of this species. So is this the same species, or is this a different one? It's something that we actually want to know. And the cool thing about this, and here, what I'm showing is just that. This big empty space is all the difference, all the genome that the marine strain has that the other strain of Ortae wernecki does not. In all of the genetic sequences and all of the genetic information, there are a lot of secrets that we want to discover. What are some of those secrets? Well, fungi, they originated in the ocean, then they went to the terrestrial ecosystems. It was a bloom of diversity, and eventually they came back to the ocean and to a lot of other ecosystems. And because of marine snow, probably they went back down there to marine, to deep sea ecosystems, and now they're present there. All of that history, all of that natural history could be coded there, where it's all of the deep sea adaptations that we don't know, that the other other fungi doesn't have, but this fungi do have. So it's very interesting. But also we need to know what's the, like, how much DNA, how much difference for gypsy fungi means that there are different species, and how much means is just the variable, the normal variables that are between or inside one species, let's say so. I'm just gonna pass this. Oh, well, this is something important, that this species of Ortae was actually the first record for the species in a regional scale. So this actually was not a new record for the country, but a new record for the whole of Latin America. And yeah, this is not the presentation that I was going to show you. So let's imagine that here says, thank you for your attention. Because here there's just more information about some other fungi, but I think there's no more time, so I'm gonna just leave it there. Thank you.
Thank you very much. Maybe we have a time for a little quick question.
I have one. How do you avoid like contamination in your sample? Like, how are you sure that your fungi come from the Dipsy and not from anywhere else?
Yes, actually, one professor from the school asked me the same thing, because we found some fungi that are in terrestrial ecosystems. So it's like, okay, so maybe they would be in the air, but we are working in a lab where a lot of other projects are happening at the same time. And if it was a contamination from the air, we would expect that some of those strains appeared in some other cultures from other people. But the strains that we found were just in the petri dishes of the samples that we took. Also, when you culture something in a petri dish, you put the sample, let's say, in the center, and if only in the center there's like fungal growth, you can be sure that it's the thing that you inoculated in the medium. But if it's like maybe in the outer parts of the petri dish, then it could be contamination. But all of the samples that we took were only in the exact spots where sediments were in the petri dish. And of course, we took a lot of care when we were like doing everything like inocuidad y esas cosas. Yes, and also we have controls like petri dishes that were just open in the in la cámara de flujo, to be like, okay, if something is growing there, it's because it's in the air, and we didn't put any sediment there, so it might be. So, yeah.
Thank you very much. Okay, our last presentation is by Emily Covell. She's currently in the final year of her PhD in the Cordes Lab at the Temple University in Philadelphia. Her research focuses on modeling the biotic niches and spatial distribution of the foundation species at deep sea methane seeps of the Pacific coast of Costa Rica.
Great. So hopefully everyone's still going. Hopefully I won't take too long. Yeah, so my work actually comes from some of the methane seeps that Jorge and Lisa Levin have already spoken a little bit about, so I can kind of skip through some of the details with some of that. But I'm particularly focusing, so this is, I'll not be too many details of methane seeps, but you can kind of consider them to be in a couple of main zones. In the active central zone of a methane seep, it's where the active seepage is coming out of the sediment, forming the basis of a chemosynthetic food web. So in these areas, you get these really nice big communities of associated foundation species. They'll often have chemosymbionts in their tissues that are utilizing the seepage fluid to kind of create biomass. And then further out from there, you have these transition zones. Those are areas that we think maybe seepage has occurred in the past, but it's since died off, and instead what you have left there are large carbonate rock formations. Bicarbonate ions are a byproduct of some of the interactions and so chemical reactions that happen at methane seeps. So they provide a really nice hard substrate for attachment of organisms such as corals and sponges. as Jorge's already talked about a little bit, this was the Rockets initiative that went and did some dives on the RV Atlantis and the RV Falcor between 2017 and 2019. My data all comes from these two cruises, 2017 and 2018, on the Atlantis. And specifically, my data comes from this piece of kit here, AUV Sentry. So, if it'll play, this is a video. Let's see if it plays or not. You might have to click on it, I'm not really sure. Normally it plays automatically, but basically it's an AUV is an autonomous underwater vehicle. So it's sort of like you can think of it like a big underwater drone. It doesn't matter if it doesn't play. It basically goes along and does two transects of the seafloor. It'll do 2 passes. So the first one, it doesn't matter. It kind of does these backwards and forwards sweeping motions. The first pass, it's higher up off the sediment, and it basically does multi-beam, so you can see the hills and valleys of the sediment. And then the second pass, it goes back down, further down, close to the sediment, and takes high-resolution georeferenced photos of the seafloor. Those photos have abiotic sensor data associated with them, so we can look at a snapshot of the seafloor and see associated data such as temperature, oxygen, salinity, and proxies for seepage. So this is the area that the cruise is focused on, and I'll draw your attention to this site here, Mount 12. That's the one I'm going to cover in this talk today. But all of the methane seeps are kind of focused right along the edge of this margin here. The subduction of the Cocos Plate is actually creating tectonic movement, faulting, that allows for the seepage to sort of come up through the sediment, forming those methane seeps. So a couple of questions that I'm going to ask in this talk. First off, I kind of went into my thesis wondering how we could maybe assess some of the potential successional stages that are occurring at these methane seep sites. So in deep sea habitats in general, things are very long-lived, slow growing, so we can't really often directly observe succession occurring. So instead, I wanted to have a look at what we know about the kind of life history of a methane seep, and then also the preferred abiotic conditions of the different foundation species of a methane seep, and see if we can parse out which are the likely successional stages. So I'm focusing on foundation species. Some of the foundation species at Mount 12, or any of the methane seep sites really along the Costa Rica margin, are down here. Foundation species we're talking about are basically, it's the equivalent of the trees and shrubs in a terrestrial forest. It's the organisms that are creating kind of substrate and refugia for other organisms. So in this case, we've got Bascomide clams, mussels, tube worms, corals, things like that. So from data from other areas, such as in the Gulf of Mexico, we've kind of passed out a few theorized successional stages, and we're looking to see if we can see evidence that supports those. So first off, I'd expect that at very early seepage, you don't have any carbonate rocks that have formed. They are going to take hundreds or thousands of years to form, so you're going to have soft sediment. I would expect that bacterial mats and clams would be the kind of early pioneer type species at an early formed seep site. Moving on from there, once the carbonate rocks start to form, perhaps mussels are becoming dominant. They require hard substrates for attachment of their visceral threads. They require active seepage to be occurring, so they have symbionts. Further on from there, perhaps tube worms are then becoming dominant. They have an extensive root system that they can use to kind of bury down into the sediments, potentially accessing deep buried sulfides that might be all that's available once the seepage kind of wanes. And then eventually, once the seepage stops altogether, seepage is quite toxic if you're not an organism that's actually utilizing it. So in that case, then I would expect non-chemosynthetic organisms, such as some of the corals, to become dominant. So the way I went about looking at all of this is through maximum entropy modeling. So that basically, if anyone's not familiar with it, you take presence points that you know an organism has occurred at, and you take their associated abiotic conditions. So from all the different sites, from AUV Sentry, I had about 100,000 seafloor photos. And we went through all of those and we looked at presences of the different organisms, pseudo-absences as well, and also some like ordinal percentage cover categories. And you basically take 70% of those points and present them to Maxent along with the like rasterized abiotic data that we got from Sentry. And you can train the model on that 70% of the data that it's seeing to start to be able to predict areas that are suitable and have suitable abiotic conditions for that organism. So you're kind of modeling its niche. And then you can test how well the model is performing on the 30% of the data that the model's never seen. And so when you do the kind of training and testing, you get AUC values. So anything above a 0.7 is considered to be a usable model. So we got really nice AUC values. You can see like 0.8s and 0.9s for most of the models, so they're performing really well, predicting the abiotic conditions of the different organisms really nicely. And there's lots of data from this, and I just wanted to highlight a couple of the more kind of the nice sort of ones for in a presentation that support the kind of succession idea. Thanks. First off, if you kind of lay these organisms out along these theorized successional stages, these predicted earlier successors all had a higher explanatory power of shell hash, which is associated with soft sediment areas. As you move along the latest successional stages, organisms become much more reliant on carbonate slabs, which only occur later in the formation of a seep, so that kind of makes sense. If you also look, so this distance to hotspot variable is a one that we calculated from SDORP. So SDORP is a variable that we can use as a proxy for seepage, but that is a kind of patchy variable, so we also calculated distances to hotspots of that variable to kind of take away some of that patchiness. And when we did that, we saw that mussels have the kind of highest peak in explanatory power from this distance to hotspot variable, meaning that they require the sort of seep to be very close to them, whereas it kind of tails off on either end. So that also, to the point where things like corals actually aren't reliant on distance to hotspot at all. And you can plot the habitat suitability of these different things too. So this is SDORP, that proxy for seepage. These more negative values along the x-axis are the most seepy spots. It kind of works backwards. Right at the center of the really seepy spots, corals really had quite a low habitat suitability, but tube worms actually didn't have particularly high habitat suitability either, suggesting that they are a later successional stage. And when you looked at distance to the seepage hotspots, tube worms and corals actually peaked in their habitat suitability at about 30 meters away from our seep. They weren't wanting to be right at the center of the seep. So again, that kind of supportive of that later successional stage. And if you look at the drop-off of the different suitabilities, mussels, as you move away from the center of the seep, mussels dropped off very quickly, so they're reliant on that active seepage fluid. So that all was pretty exciting. That was all data from 2017. But something threw a spanner in the works slightly because there was a large earthquake that happened in the area at the end in November 2017. So I wanted to see if there'd been any clear effect that may be attributed to that earthquake. So this is the area where the earthquake occurred, and this is Mount 12 here, so not very far away from it at all. And this is the SDORP, so these, this is 2017 and 2018 here. These kind of pink and yellow areas are the seepage areas, and you see that in 2017 you've got this line of seepage at the established active site of Mount 12, which has kind of disappeared in 2018, and now the seepage is right around the periphery. And if you're a sessile organism like a mussel or a tube worm that's reliant on that seepage, if that's a permanent thing, which we don't know, but if it is, that's a bit of a bummer. So we ran the models again, looking at the AUC values. The AUC values were always lower in 2018, so the models were struggling a lot more. When you look at the habitat suitability maps, it starts to make sense why. So this is 2017 here, and 2018 here. This is the one you just saw, which had these nice clear patterns. The pattern is now a little bit crazy in 2018. Same for distance to hotspot. You had these nice clear sort of peaks here, and it's now gone a little bit haywire again. So that kind kind of makes sense if that seepage has moved and the organisms aren't actually moving to keep up with it because most of them are sessile. Perhaps something, now these organisms aren't actually in an ideal abiotic condition. So the final thing we did, we focused in on the overlapping areas where we know we have photos that we can basically say were taken on the same spot from one year to the next, and looked at those percentage cover values of the mobile organisms, so the clams. We have two different species of clams that we think we were seeing. The first one are Archivesica clams, which are the ones that form those nice big clam beds. And so we mapped the changes in Archivesica percentage cover against the changes in seepage conditions. So the background squares are changes in conditions, so the red squares are areas where seepage increased between the years, and it actually correlates really highly with changes in clam coverage as well. So that's potentially suggesting that as the seepage has moved, the Archivesica clams have actually shifted and followed the seepage. They're now established clam beds in these new seepage hotspots. So that is kind of further evidence that they're possibly, the first successional stage of the new seep site and that kind of pioneer species. And you had the opposite pattern for the Thyasira clams, have a kind of different life history. They live buried within the sediment. In 2017, you didn't really see them on the photos, whereas in 2018, there was loads of them on the photos, and they had clam tracks kind of going long after them on the sediment, suggesting that they're moving across the sediment actively. And they had the opposite pattern. So in areas where seepage decreased between the years, you saw a real increase in these very mobile clams. So thank you. So it's possible that that's actually like a disturbance effect. In areas where the seepage has decreased, these clams have now come out of the sediment and maybe actually kind of trekking around trying to find more suitable conditions. So just to conclude from this and from a lot of my other work, in my thesis we are seeing support of these succession patterns, so that's really cool. We're actually having a look to see if we see some of these patterns at some of the other methane seep sites. So that's kind of a little bit of a work in progress. I'm still writing that up. But yeah, and thanks to everyone that's been part of this research. And if we have time, I'll take some questions.
Thank you so much. Maybe we could have a time for one question. Yes, okay. Questions? Okay, I think, okay.
It's basically a scientific illustration kind of program. It's got preloaded, you can load your own vector images into it if you want to, but it's also got a lot of preloaded things that are kind of fun. It's really cool for microbiome kind of stuff as well. There's a lot of things in there. I can send you the link to it.
Okay, thank you very much. So.
Thank you for staying until the end. Now we'll move to another lobby of the CIMAR Research Center, so we can walk, we can show you the way. And tomorrow we'll be here at 8:30 with a coffee, but we start the presentations at 9. But if you want to have coffee here and chat with the rest of the group, you're more than welcome. And yeah, thank you for everyone for an amazing day. And we'll see you with a brindis over there. Thank you. Also thanks to Giancarlo, who's been there in the back.